Good evening, everybody. Thank you for joining us this evening. I really appreciate you all taking the time to be here at the Koch Institute. I'm going to briefly introduce myself. I'm Jane Wilkinson, the Executive Director, and it is my privilege to introduce this amazing panel of speakers that we have this evening. We're going to start out with a fantastic introduction of what immunotherapy is and how immunology actually has a huge impact on cancer, its spread, and also the treatment. So I'm going to introduce Stephanie Spranger. And Stephanie, you can take it away and tell us all about immunology.
Thank you, Jane, for this introduction, and welcome everyone to the KI. It's my pleasure to introduce immunology to you and how we aim to use the immune system to target cancer. Just to illustrate how powerful the immune system is, I wanted to start out with a movie where we actually see a cancer cell dying after contact with an immune cell. And in this case, you see here the immune cell being the pink cell over here. And specifically, this is a killer T cell. And this big turquoise cell is a cancer cell. And you see here over time, the turquoise cell dies, it shrivels away only by the virtue of this contact with this immune cell. Immunotherapy is not necessarily a new invention. The first person that attempted to use the immune system to fight cancer, and I should say not just attempted, but actually clinically did this, was William Coley. William Coley recognized that if we use a mix of bacteria, heat and activate this mix, and then inject it straight into the patient's tumor, we can induce complete regression of the tumor. And I want to point out this was 1890, so over a century ago. However, because of the ill-defined composition of the bacterial mix that he was using, this in the 50s, around the 50s, did not become mainstream for the treatment of cancer anymore. It took almost a century to reintroduce immunotherapy into the clinical setting. And this was done by, at the time, the director of the surgery branch at the National Cancer Institute, Steve Rosenberg. And he recognized that we can surgically remove cancers from patients. And he specifically removed only one cancer lesion from a patient that had multiple cancer lesions. Then isolated the immune cells, these blue cells within the tumor mass, expand these cells in an in vitro setting to very large numbers. And he used one trick that will become important throughout this talk, which is he added something that makes these T cells proliferate to accumulate in large numbers, which is called IL-2, that's a cytokine. And then he reinfused these killer T cells back into the patient. And just to give you an idea of what happens to those patients, this is one radio image of a patient. Here you see the lung, and here you see two large tumor lesions, metastatic lesions of that patient. Within the span of not 2 months, those cancer lesions went away. So what I just showed you in the movie, that those pink cell can kill this one large turquoise cell, this can also happen in patients in real life. So how do our killer T cells achieve this goal? Every single T cell that you have within you has a unique receptor. And this unique receptor gives this T cell the ability to recognize what we call antigens. And anything those T cells will see, they do in a very specific manner. So it has to be a perfect match between the T cell and its antigen expressing cell. And in this case, here illustrated, this T cell recognizes an antigen on the cancer cell and will then induce cytotoxic lysis. However, we have to recognize that it would be incredibly dangerous if our T cells could just walk around in us, crawl through us, and trigger when they see an antigen that they're specific for, because then we would have an enormous amount of autoimmune diseases. So what has to happen is that we have a licensing system where the T cells become licensed to then acquire this killer capacity. And the cell that can induce and instill this license to the killer T cell is called a dendritic cell. This was first discovered by Ralph Steinman, and it was called dendritic cells because these are microscopy images of the cells. They look like trees. They look like beautiful cells. And these can differentiate. They have the unique ability to differentiate between danger and no danger. So just to give you two examples, in the top example, these dendritic cells recognize that there potentially is a viral infection or a bacterial infection. They trigger and they will induce immunity. So large proliferation of those killer T cells, which can then patrol and eliminate diseased cells downstream. And in contrast, the same dendritic cells can recognize that down here, we don't have danger. That could be, for instance, a food antigen that you eat. The dendritic cells recognize this is no danger to our body, and these will induce tolerance. And that is why we, for instance, don't overreact to food antigens. So how does this work when we put it into the cancer context? At the beginning, those dendritic cells recognize that there is danger, that there are diseased cells that are under stress, and they will sense that and then pick up antigens from those tumor cells and carry it over to the closest draining lymph node. And now I have to explain to you what a lymph node is. Lymph nodes basically are data hubs or meeting points for immune cells. So it's an organ where many immune cells come together to increase the likelihood that the right dendritic cell meets the right T cell. If that is the case, and we have a specific antigen interaction, we get activation of those T cells, and these T cells proliferate to large numbers, acquire effector function, which allow them to kill, and then migrate back to the site of the tumor and start eliminating cancer cells. And that's what we call the cancer immunity cycle. However, if this cycle would work perfectly, as I just described it to you, we would not have cancer as a disease because all cancer cells would be effectively eliminated. What happens, unfortunately, over time is that the T cells literally become exhausted. They reside in the tumor and they fail to exert the killer capacity. And this is where the current form of immunotherapy, the most frequently used immunotherapy, actually acts. So in the normal scenario that I introduced to you earlier, the T cells can kill. And over time, a lot of immune inhibitory signals that the cancer cells and other cells in the environment put on the T cells make those T cells exhausted, and they can no longer fulfill this killing function. And we now have drugs that block the inhibitory signals. That allows the T cells to regain the killer capacity and start eliminating cancer cells. That this actually works in the clinic and has changed how we think about many cancer diseases can be illustrated by this clinical data. And I drew in here the historical data on how chemotherapy would look like. And the main point here is at the end of this time span, very few patients are alive. And in contrast with immunotherapy, and this is several types of this immune checkpoint blockade immunotherapy, we get about 50% of the patients to have long-lasting, very durable responses following therapy. And this works not just in melanoma, that was the cancer type I just showed you. It works across several other cancer entities, but it only works in a fairly small fraction. So these are the best responses across cancer types. And you can see there's many other cancers out there where immunotherapy currently is not effective. So bringing back what is needed to drive an anti-tumor T cell response. We need three main signals that those dendritic cells instill to activate a T cell response. The first is here illustrated as the antigen. That gives the T cell the specificity. The second one here is a positive or a negative signal called a costimulatory signal. And this is where immune checkpoint blockade therapy attacks this immune response. And it shifts it back to more positive signals, enlarging the T cell response and giving it more effective function. But then we also have a third signal down here. And this third signal can be summarized as cytokines. These cytokines basically give the immune response the flavor, the color of what the functional state of this response is. So either a red or a green response, if you will, and how durable this response is. However, I drew this in here with one single example. This is much more complicated if we layer this onto the cancer immunity cycle, because cytokines are a very large class of molecules. They're soluble and they're very variable in their amount, abundance, and where the T cells will encounter them. So which cytokines, where, how, and for how long is incredibly important as the T cell will go through this activation cycle. And just to give you three very specific examples, the cytokine interferon has to be perfectly dialed to make this immune response work. We need it both in the lymph node as well as in the tumor, but too little or too much will break this immune response. As a second example, the two cytokines IL-2 and IL-12 we know are essential to drive this expansion in the lymph node, but too much of them will induce autoimmunity. And lastly, we also know that TGF-beta in the tumor microenvironment will provide an inhibitory signal to the T cells, but without it, we likewise would have an incredible amount of autoimmunity. And with that, I'm passing it over to Dane Wittrup, who has dedicated his career to understand and use engineering to modulate the cytokine signals.
Thanks, Stephanie, for that introduction, setting the stage. So with the promise of these cytokines to rev up the immune system to attack your tumors, initially back in the 80s, what people did was they produced these cytokines and just injected them into the blood of patients. You just soak the person in this alarm signal. And what happens is their entire body reacts as if it has a fulminant infection. And so the toxicity is considerable. In fact, for some of the cytokine therapies that were tried first, you had to be in the intensive care unit for a week or two while you were being treated because your whole body was under this state of alert. So one of the obvious ways you might fix this and come a little bit closer to the actual way that cytokines are deployed by our immune systems would be to limit the cytokine to the tumor where you want the activation of the immune system. And dozens, or maybe at this point, hundreds of clinical trials have been done where people injected such things directly into tumors, which is a good start, but unfortunately they diffuse right back out. Because given the characteristics of tumors and their leakiness, you may gain a short period of time where your treatment is local, but it rapidly becomes systemic, which then limits how much you can deliver. So what you're going to hear more about in the rest of tonight's talk is attempts to put these cytokines in place where you want them and make them stay there, and different ways of doing that and ways we hope to exploit that. Because if you do, then you can get the localized immune response that you're trying to generate without being limited by the overall reaction of your entire body. So first, I'm going to tell you a bit about one way of accomplishing this, which is something that a former grad student in my lab, Noor Momin, developed. She'll be starting her own research lab as a faculty member at the University of Pennsylvania next year. And what she proposed to do in her thesis work was to use existing collagen in tumors because it's just about everywhere, including the great majority of tumors. There's a lot of it, and it stays in place. So it provides potentially a point to grab onto. And the way she did that was through recombinant DNA, expressing a collagen binding protein as a piece of one fusion protein to the cytokines. So now the cytokine is given the functionality of grabbing onto collagen. She also put in an albumin molecule here, which just made it big enough so that it didn't diffuse away before it got a chance to grab onto the collagen. And here's some of the images that Noor took of some of the mouse tumors that we treated, showing where the collagen was. There's lots of different types of collagen here. It's just showing a couple of them. It's in a sort of a capsule around the tumor, sort of a scar tissue. It's also around every one of the blood vessels in the tumor. So these are places within the tumor that the cytokines would be held. And so here was some of her characterization of how this worked in a melanoma tumor transplanted into mice. This first plot here just shows that it stayed mostly in the tumor. It's a log plot. In other words, they go by powers of 10. So basically, over the course of several days, the cytokine in the tumor is at 10-fold higher levels than it would have been if we didn't try to retain it. So that's good. We increase the amount that's in the tumor. On the flip side, if you look out in the blood, there was 10-fold less of the cargo out in the blood. So we accomplished both things we were trying to do with this collagen binding. Kept it at higher levels in the tumor, lower levels outside, which then caused the mice to lose less weight. Sort of the zero order measure of how sick these mice are is if they're losing weight. We can get in far more molecular detail about how they're reacting to these, but they lose quite a bit of weight. 10%, picture, losing 10% of your weight in a few days, you have to be pretty unwell. So if we didn't retain these cytokines, and in this case it was IL-2 and IL-12 among the ones that Stephanie talked about, they would lose 10% of their body weight. But if we retained it inside the tumor, they didn't. And what this let us do is cure these mice of the tumors that we had given them by implanting them. So the blue curve here is a particular dose of those two cytokines, 2 and 12, not trying to retain it in the tumor, injecting it into the tumor, but not trying to make it stay there. All the mice died. They lived a little bit longer, but they all died. If we added the collagen binding domain so that it didn't diffuse out, over half of the mice were cured. So it's sort of a almost all or nothing difference. So with that, I'm not a medical doctor, but I am an engineer, and so we're goal-directed. We're trying to make things to have impact in the clinic. And so it's a long way between what we were doing with these mice and humans in the clinic. Long way in terms of time, in terms of resources, in terms of effort, in terms of money. So hoping to find, I didn't think that was a long time. Hoping to find something intermediate that we could still learn something and advance these programs. We started a collaboration with Tim Fan at the University of Illinois, who has a program he calls Pets to People. And these are not models of cancer. These are pet dogs with cancer. And so what Tim Fan and my group started doing was a clinical trial on dogs with oral melanoma. They get melanoma mostly in their mouths because the rest of their body's covered by fur, so it's protected from UV radiation. And what we would do is give them a shot of radiation on the tumor initially, and then go through 6 cycles of treatment with IL-2 and IL-12 engineered to bind to collagen. So just gonna warn you, these are pictures of tumors in a dog's mouth. So if you don't want to see that, I'll telling you that that's what's coming up on the screen next. It goes away. It's good news by the end of the slide. But anyway, here's the initial bad news, which is at this point, this dog, this was inoperable. That tumor, over half of the mass of that tumor was actually back up in the skull. So this was inoperable, day 1. But after 2 doses, you can see it's become much smaller, and it's also no longer 2 colors. And then you go on out to after all 6 of our doses, and it's disappeared. And now, coming up on almost 2 years later, that dog is still running around in the owner's backyard. So that's an anecdote. That's one dog. There were other dogs with similarly dramatic responses. And if we look across 13 of the dogs that we treated this way, about half of them, their tumors went almost entirely away. And this is in the term of art, this is called a waterfall plot. And so that is exciting and inspirational for us. But this collagen binding is only one way of making the cytokine stay in the tumor. I'd first heard about this field actually from my colleague Daryl Irvin, who had used a different way years before with his student Brandon Kwong, attaching cytokines to nanoparticles in tumors. And so then subsequently, Daryl and I co-advised a student, Yash Agarwal, to use yet another way to try and localize cytokines in tumors. And that's what you hear about for the rest of this evening's talks, using a biomaterials approach. And so to introduce briefly Daryl, one of the best things for me about being in the Koch Institute here at MIT is being next door to Daryl. He tries to get work done, but most times when I walk by his office, there's something I would love to just stop and talk to him about. So you just got to close that door if you don't want me coming in here. But Daryl is one of the world's experts on designing creative new ways of exploiting biomaterials to make vaccines. and also for exploiting biomaterials to make new immunotherapies for cancer. So at this point, he'll pick up the talk and carry it from there.
Okay, thanks, Dane. So how the story evolves is about the same time that Dane's lab was exploring this collagen binding approach to treating cancer. Half my lab works on vaccines for infectious disease and half the lab works on cancer. And there are many, it's not infrequent that the work we do on one side of the lab affects what's happening on the other side. And this is an example of that, where while Dane's group was exploring these collagen binding immunotherapies, we were trying to develop ways to improve subunit vaccines. And so from the vaccine side of the lab, we were working on this problem that when we administer a vaccine, the simplest kind of a vaccine is where you take a protein that we call the antigen, which is what you want the immune system to target. If this was SARS-CoV-2, it might be the spike on the virus surface that causes it to bind to host cells. This is what we want to target with our immune response. But just injecting that protein is not going to elicit an immune response. We have to mix it with an adjuvant, which is a generic term for something we mix with the antigen that will get the immune system to pay attention to it. And adjuvants often are meant to induce a little bit of mild inflammation that brings in immune cells and gets the immune system reacting to what we've injected. The oldest vaccine adjuvant, it's been used in humans for 100 years. It's called alum, and the material is aluminum hydroxide, and it comes in a vial. It looks like a milky solution. And what alum is it's an aqueous suspension of aluminum hydroxide particles, basically tiny metal particles. That it was discovered in the 1920s. If they were mixed with an antigen and injected, they would cause some very mild irritation in your tissue that would get the immune response to generate strong antibody responses. And what we were looking to do is come up with ways to make alum more potent, because alum is very safe, but it's known that it has a limitation in how strong the immune response it elicits. And so we're always interested for infectious disease, can we get stronger antibody responses, stronger T cell responses? So the approach we took to improve the potency of alum was to come up with a way to try and anchor our vaccine antigen onto the alum particles rather than just mixing them together and injecting them. We wanted the antigen to stay bound to the alum particle in hopes that it would stick around longer when we injected it and better simulate the immune response. And here we were able to exploit some chemistry where we knew that if we had a phosphate group on our antigen, it could actually react with the surface of these alum particles and chemically stick it onto the surface. And so we came up with a strategy of introducing these phosphoserine groups, which are just peptide residues that would have a phosphate and react when we mix the antigen with the alum and keep it bound. And what this does is that when we go and inject this into animals, if we just mix the antigen and alum and inject them, the antigen here that we've labeled so that we can image it in the live animals goes away from the injection site very quickly within about a couple of days. But if we use this phosphoserine chemistry to anchor the antigen on the alum, now it sticks around much longer. And here we're quantifying that in the animals. So the antigen just mixed with alum, it's gone in about 3 days. But if we use this chemical attachment, now the antigen sticks around much, much longer. And that has a tremendous impact on the strength of the immune response. You're just basically giving the immune system more time to react to the antigen. And so, for example, now if we use this approach with an HIV antigen and measure neutralizing antibodies against that HIV virus, we go from a very weak response where half the animals make no neutralizing antibody to a much stronger response where all the animals are making strong neutralizing antibody. So this was great, but Dane and I are talking. He's again coming in my office all hours. I don't get any work done. And he was telling us about the collagen binding, and we wondered if this technology might actually be a really interesting modality to adapt to cancer. And the idea was kind of obvious from what you already heard from Dane. What if we could take our cytokine drugs for immunotherapy and use the same phosphate anchoring approach to stick these onto alum. And now we could inject this into a tumor and use alum as the anchor rather than collagen. And there are a couple of reasons we thought this might be useful. Collagen is ubiquitous, but it might vary patient to patient or tumor to tumor. Here we can sort of know what our anchor is, and this can really sort of perfectly hold the protein here. Dane showed that the collagen binder, the protein sticks around a few days. Here, as I'll show you, we can make protein stick around for weeks. So now if we bind our cytokine IL-12 to alum and inject it into a tumor, the alum goes throughout the tumor volume, and so does the cytokine, and they're on top of each other, they stay bound. And if we image in the animals the signal from the cytokine, if we inject free cytokine in the tumor, it goes away very quickly. But if it's bound to alum, it's sticking around days and days and days. And this had a pretty big effect on the toxicity. So if the IL-12 cytokine is bound to alum through this chemistry, you don't see any of it show up in the blood. But if you inject the free IL-12 here in orange, a few hours after injection, it's all going out into the blood. And that leads to this weight loss that Dane mentioned, where the animals are getting very sick as the drug leaks out of the tumor. And in mouse models of melanoma and breast cancer, this had a pretty tremendous impact. So if we dosed melanoma-bearing mice with IL-12, they didn't survive much longer than being untreated, but a single shot of this alum-bound cytokine cured half the animals. And this is a model of breast cancer and a similar sort of story. The animals all succumb if we inject free cytokine, but with that drug bound to alum, they reject the tumor. So this worked so well, and we tested it in many models. And to connect back to Stephanie's talk, what we really think we're doing is we're helping the T cells see that cytokine signal that helps them be really good tumor killers. And so at this point, Dane and I both felt that we tested this as well as we could in the lab, and it was time to move this out and try and move it toward the real world. And that's where our last speaker, Howard Kaufman, comes in. And so let me introduce Howard. So Dane and I started a small biotech company called Ankura to move this technology out of the lab and into patients. And Howard is the leader of Ankura. He received his MD from Loyola University and did a residency in general surgery at Boston University, fellowship training in tumor immunology and surgical oncology at the National Cancer Institute. He's been a director of surgical oncology at the Herbert Irving Comprehensive Cancer Center at Columbia. He was the director of the Rush University Cancer Center, an associate director for clinical sciences at the Rutgers Cancer Institute. He's really been a leader in immunotherapy for a long time. He led the phase 3 trials of the only FDA-approved intratumoral therapy called an oncolytic virus, and also led pivotal clinical trials of a checkpoint blockade antibody, one of the other immunotherapies that you've heard about. He has also then moved from academic roles to work in the pharmaceutical industry to help move things toward patients. He's worked as a chief medical officer at Compass Therapeutics and Replimune, and was most recently the head of research and development at the Immuneering Corporation. So when Dane and I started Ankura in 2020, and we heard that Howard could possibly be interested in a new position, we were immediately really excited. Howard joined Ankura in 2021 and became the CEO in 2022. And so please, let's welcome Howard to give his perspective, both as a clinician and as someone trying to move therapies into patients.
Thank you, Daryl. So I am a physician, and actually I take care of cancer patients. And my first personal interaction with the disease was actually when I was an undergraduate, and my grandfather had cancer. And, you know, in those days, my grandmother didn't want anyone to tell him. And when we were alone together, he said, you know I have this thing. And they said, there's nothing that can be done. And when I got to medical school and I started learning about what cancer really was, and that we often didn't have treatments for patients, it was really frustrating. So I decided what I wanted to do is take care of cancer patients, but I wanted to bring them something new. And And so I knew that to do that, I had to go to the lab and go to the basic science to get that. And at that time, you really were a basic scientist or you were a clinician, and wanting to do both was not very popular. But fortunately, the NIH actually gave a word to this, and they called it translational research. And academic centers, whether they wanted it or not or knew what it was, I don't think so in most cases, but they knew they wanted to have somebody who did that. And so it's kept me employed for a while. And I thought I'd share my story about how I got into this particular area. So when I was starting out in the lab I went into had been involved in cloning genes. And for those of you who are students and young, cloning a gene back then could take a few years to actually do. So it wasn't something that took 8 hours. And the lab I was in had cloned the gene called carcinoembryonic antigen, or CEA, which was a tumor antigen. And in fact, my first job in the lab was to do immunostaining to see what tissues had the CEA in it. And one day I asked my boss, I said, couldn't we use the CEA and immunize a patient to get an immune response to the cancer? And I remember what he told me was that he said, Howard, that is so obvious. Other people must have done it and it didn't work, or everybody would be doing that. So I was dejected and I went back to the lab and talked to Judy Cantor, who is the main person I worked with. And I said, what do I do now? That the big boss said, I can't do this. And she goes, what we do now is we ignore him, we do it anyway, and then we show him the data. So that's what I did. And what we decided to do was that to immunize the patient against the CEA, we couldn't just give the CEA, we had to give it in some way that the immune system would think it was a problem. And so there was a guy at the NIH who worked across the way, a guy named Bernie Moss, and he was a pox virus expert. And he had learned how to put genes into the pox virus. And so I actually put CEA in vaccinia, which at the time was the very first eukaryotic gene to ever be expressed in a virus. And we published that, and I spent some nights in Bernie's lab figuring out how to do this. And then I actually had the opportunity as a resident to run the very first phase 1 study of this in patients. And the good news was at that time, the FDA was very worried we would kill everybody because we're giving them a virus, and we thought these patients were immunosuppressed, although I think that definition has changed today now that we understand the immune system better. And what happened is that it was actually quite safe. We had really no side effects to really speak of, but it didn't really work. And so then I got my first job and I began to think about why didn't it work? And I thought, well, one thing that we did show in this was that we were getting CEA specific T cells. So as you've heard today, T cells are really critical to getting rid of cancer. And so when I thought about, well, how did these patients get T cells, at least that we could detect in the blood, but their cancers weren't going away? And at the time, the thinking was, well, maybe something was happening to those T cells when they got to the cancer and they couldn't do their job. They were getting immunosuppressed. So then the question became, what if we just went to the cancer directly? And we kind of overcame whatever the local problem was. And so the way that I decided to approach this, actually my very first NIH grant, was to take the vaccinia virus and inject it into the tumor. And by that time, we had learned some of those co-stimulatory molecules that Stephanie spoke about. And the idea was, what if we delivered that with the vaccinia virus directly into the tumor? So when I started my research, I was going to be a colon cancer doctor because CEA is mostly in colon cancers. And when we went to the FDA and I said, can I inject this in colon cancers? They said, why don't you do melanoma first? It's on the skin, it's easy to get to, it's more immunogenic. So I said, okay. I wrote a colon cancer grant to the NIH and I wrote a melanoma grant. And well, you can guess which one got funded because we did the study in melanoma. And this actually was one of the very first patients I ever treated in New York. She had advanced melanoma. It had started in her foot. That's actually a skin graft site on her leg where the original cancer was. It then spread to her lymph nodes and her lungs. And we treated her by injecting one of the lymph nodes in her groin. And what happened is after 4 vaccinations, all her cancer went away. And shortly after that, she developed this whitish patch around her mouth and in her on the skin graft site, which is vitiligo. And the reason that develops, we now know, is because the immune system is not only killing the melanoma cells, it's killing the normal melanocytes. And when we measured her T cells to several melanoma antigens, one called MART1 and one called GP100, you can see before treatment, there were a few of them there. After 3 months, not much difference, but then at 6 months, boy, she got a whopping response. And I followed her for 5 years and then lost track. She, as far as I know, never had her cancer come back. And that's what really started my career. And then what I realized is I had accidentally developed what was an oncolytic virus, because when we put the virus into a cancer, the viruses tend to actually kill the cancer cells in addition to generating an immune response. So my wish to be a GI doctor was gone, and I became a skin cancer doctor, which was fine. And as it turns out, the skin is actually the largest organ in the body. And so not surprisingly, it's the source of most cancers in the world. About 20% of Americans will develop a skin cancer at some point in their life. And the sad statistic about this is, although we tend to think of it not as a serious cancer, and most of them are not serious, but they can be serious and they can spread. And sadly, 2 people die every single hour from a skin cancer, mostly actually today squamous cell, but also melanoma. And the non-melanoma skin cancers are going up at a very alarming rate of as much as potentially 77% on an annual basis. So a lot of this is related to the sun. Probably also people are living longer, and we know that being immunosuppressed is a problem. So this is a real issue in like patients who've had transplants. And of course, this is extremely costly to deal with this disease. Now, when I started my career, we actually had 2 drugs available to treat melanoma. One was dacarbazine, which is a chemotherapy. And interesting statistic, it was never ever approved by the FDA. Just as people were using it, probably wouldn't have passed FDA approval. In the literature, it had response rates anywhere from 5 to 30%. Highly unreliable as a control drug in a study because of that. But no matter how well the patient responded initially, almost all of those patients eventually recurred, and it really didn't have a survival benefit. And then interleukin-2 got approved. That was the first, really the first major drug for treating metastatic melanoma. It was actually first approved for treating kidney cancer in 1992, and then a few years later got approved in melanoma. Has a response rate of about 15%, but it could be curative. In fact, some of my longest surviving patients were IL-2 patients. I have one now who's now over 18 years, and once a year on his anniversary, we go out to dinner. And he's actually ended up chairing the Melanoma Research Foundation, which was very nice that he decided to give back. He was getting his affairs in order, and I said, you know, before you do that, let's do this treatment and see what happens. And fortunately, he had a complete response. And being a melanoma doctor back in the late 90s, early 2000s was really easy because patients either qualified for IL-2 and got it or they didn't. And that was it. And then in 2011, everything changed. And the checkpoint blockades were first studied in melanoma, and the first drug, ipilimumab, got FDA approval. And after that, PD-1 came along, and PD-1s are now approved in 16 different cancer types. And I think a total of like 78 different sort of indications across the board, and really completely changed the field. And melanoma went from a drug, we used to go and beg drug companies to study melanoma. Nobody wanted to touch it because nothing ever worked there, to all of a sudden melanoma has become the poster child, because not only did it really usher in the new era of immunotherapy, but on the top of that, it also ushered in the targeted therapy era, because about 50% of cutaneous melanomas have a BRAF mutation. And it turns out that BRAF/MEK inhibitors have a 70% response rate in metastatic melanoma. And so we went from having basically 1 drug to having now, we have 14 drugs approved. And it's a great time to be doing melanoma, but clinics now take a lot longer. In the middle of all of this, the first oncolytic virus was being studied. And because of my previous work, I got a call one day to ask if I wanted to do an oncolytic herpes virus. And I said, sure, why not? And we studied that. And that became the long name Telimagene laherparepvec, or we call it TVEC for short. And that also got FDA approved in 2015 in the middle of all of this, and really became the first oncolytic virus approved. And there's actually a second virus that just got approved in December of 22 for bladder cancer. And it's a non-oncolytic adenovirus that expresses interferon alpha. And the way that oncolytic viruses work is that they infect the tumor cells, as shown on the bottom there, and it kills them. And it does a number of things. So first of all, it induces that local inflammation that we've heard about today, which is a really good thing. It also causes release of the tumor antigens, which can then be used to essentially immunize the patient against their cancer. And importantly, with oncolytic viruses, every tumor cell that's killed produces about 10,000 new viral particles, which can then infect other tumor cells, and it becomes kind of an amplifying drug effect. What happens if the virus gets in normal cells? Well, normal cells have antiviral machinery factors. Probably the most notable one is interferon, because it interferes with viral replication. And so those cells actually can clear the virus, and it doesn't really affect them. So that's the sort of way that oncolytic viruses work. And if you haven't seen that before, this was actually a patient I treated a while back in New York. He actually was a retired judge. He went out on the golf course and then got melanoma. It started in his toe. It then started spreading up his leg, including in his lymph nodes. He actually had been treated with chemotherapy and it didn't work. And so we injected that one near the ankle and the back, and we didn't inject the others because we wanted to see what would happen. And what happened is after about 3 injections, we began to see a flattening of the lesion. It begins to crust over. And if you look around there, the other lesions are also starting to go down as well, even though they weren't injected. And over time, he basically went on, had a little bit of inflammation left. We did biopsies and he had a complete response. He's another patient that then went on and really had no significant problems as far as I know after that, went back out to the golf course. And importantly, the side effects we saw with this were really minimal. So we saw a little bit of fever and chills for about 12 hours after the injection, and then a little bit of pain or redness at the site. That's about it. And when we get the T cells out of this patient, again, going back to the video that Stephanie showed you in the beginning, these are melanoma cells that are stained in green. That's a green dye. And the blue is a CD8 T cell that receptor sees MART1. And after the treatment, what you can see is those blue cells, when they touch the melanoma cells, they're actually killing them. And the T cell itself is still alive and can then travel elsewhere in the body and kill those melanoma cells wherever they may be. And so the importance of this is that the local injection of an oncolytic virus is now FDA approved. It's been validated as therapy. It's very safe, and it induces an immune response. And this really opened up this whole field of local therapy for cancer. And so you've already heard the story about how using aluminum hydroxide to really get these cytokines into the local tumor site is possible. And that really came out of the basic labs here, and is what Ankara is now trying to take forward and put into patients. And our lead asset is interleukin 12. And why did we pick interleukin 12 to start on? Well, as you've already heard, it has a role to play in cancer. And actually, when I was a fellow at the NCI, we had actually done some studies where we gave IL-12 intravenously, again in melanoma and kidney cancer patients, and it actually worked. That we saw patients that had responses, but 2 of the patients died, and they died of liver toxicity. And because it was so toxic, it basically didn't get developed anymore. So if the underlying hypothesis that you've heard about today is correct, that we can give it locally and we won't have the side effects, it really fits a good example for that, because we already know that it kind of works, at least at higher doses. The other reason we picked it is when we studied it in the lab in the mice, it works really, really well. And so what we do is we link it on to the aluminum hydroxide. It's called anchored, and the Greek word for anchored is Ankara, which is where the company comes from. And although I wasn't really planning to move to a new company, once I actually got to talk to Dane and Daryl and the group here, it was impossible to turn that opportunity down. And so just to show you One of the reasons I got so excited about this is we've done some studies as well. And so this is a mouse, and the mouse has a tumor in the flank that's been established. And in this particular experiment, we're just injecting the regular IL-12, which also has the little bit of the alum linker peptide. And what you can see is within 15 minutes, there's a lot of drug there as shown by the red. That's the drug in the tumor site. And at 6 hours, 90% of the drug is already diffused away and is gone. At 24 hours, it's completely gone. Now, what happens if we anchor to the aluminum hydroxide? Well, we can still detect drug almost 30 days later. It's still there, and there's still some of it left behind. But what that translates to in terms of a treatment effect is what's really remarkable. One dose, not a dose every week, not 3 times a week, not every day. One single dose is the red line there with the anchored IL-12 versus the unanchored IL-12 in blue. All of the tumors went back down, and this led to survival in the animals, and they seem to be then protected from this kind of cancer. And so this is quite exciting. We're gearing this up to go to the clinic. As you can imagine, there's a lot of work that needs to be done to get to the clinic. So we're hoping to submit our what's called an IND to the FDA in September of this year. Prior to that, I had heard the stories from Dane about the dogs, and I had seen the picture that he showed you. And that got me really interested. And it turns out that one of our employees at Ankara was a student in Dane's lab and just happened to, I don't know how the story goes about how we actually had the canine IL-12. But anyway, it turns out Ankara had a canine IL-12. And I asked Dane if he would introduce me to Dr. Fan at the University of Illinois. And we said, could we do a study? And so this is our study design, which we designed exactly as we were proposing to the FDA to do it in humans. So we took dogs with melanoma, and if they're screened, we did what we call a dose escalation, where we treat 3 groups of dogs at 3 different doses. Shown here, we give it every 3 weeks for 4 times. We do a CAT scan, and if dogs are doing well, they can have that repeated. Good news, no dose-limiting toxicities, no grade 3 toxicities at all. All that we've seen is a low grade, what we call a grade 1, local inflammation at the site. And one dog had a little bit of an increase in a liver enzyme that went away after a day. This is the kind of side effects we're seeing. This is the golden retriever with melanoma. You can see this dog has multiple metastatic melanoma lesions. The red arrows were ones that were injected. The blue were not injected. You see there's a little bit of redness there. That was the only side effect this dog had. It only happened after the first dose. This dog was treated 9 months ago and is still alive and basically doing well. This is another dog. I know you've seen the horrible melanoma pictures. This is a dog treated at the intermediate dose. It has a melanoma on the hard palate, which is a very difficult location to have a tumor. And after 2 doses, this tumor started going down. This dog has now had what we believe is a complete response. It's actually continuing on the study as 1 more dose to get, and we're quite excited about what we're seeing so far. We've done biopsies before and after. This is red that is staining for T cells at the site, and you can see that there's very few, if any, before the treatment, but after 2 treatments, not only are there a lot of T cells there, but they're beginning to form real structures there, which we think are very important for fighting the cancer. We're also, you've heard today a little bit about the dendritic cells and macrophages. So IBA1 is a stain for dog myeloid cells. And again, there's none at the beginning, but you can see they're really coming into this tumor site after only 2 injections of treatment. And finally, we're seeing increase of what's called PD-L1. That's one of those checkpoints. And there's now a new canine anti-PD-1. So we're actually in discussions with the company that's making that drug, which is Merck, to do a combination study. And we're actually meeting with them next Tuesday to actually potentially design a study of the combination in dogs. We're developing other things behind us. The company is growing. In fact, our newest employee who's accepted is a MIT postdoc here. So, you know, anyone in the audience who might be in a position of looking for a job in biotech, you know, please let me know. We're hoping to develop a lot of new products. And I think this is a really good example of how important the relationship is between industry and the great work that's being done at MIT. finally, Ankara is going to be successful, not only because we have good science, but because we have great people, and we've been building out our teams. And Mike Schmidt, who's our Chief Scientific Officer, was also worked with in Dane's lab before, and just continues our close relationship with MIT. Thank you.
Thank you. We're going to move to the front and take some questions. Is there a question from the audience, or shall I get us going? I'll start. Oh, great. Saves me the job, although I have to walk.
This may be a dumb question. I mean, in the dog study that you had mentioned originally, I mean, they started with radiation. And I know a lot of the immunotherapies were used in combination with chemotherapy. What mechanism, how do you think, how do those interact? How does that improve the effectiveness? What's the mechanism?
There's a body of literature. There are a number of publications showing that potential interactions would be the radiation kills some cells, generates some antigen, and also drives inflammation. And so if you phase things right, you can have it be a part of that cancer immunity cycle in generating something for the immune system to see and beginning to bring the immune system in. It's not. It's not that simple, because if you keep hitting it with radiation, you kill the immune cells that are coming in. So it's tricky in how you actually do it.
You know, the original abscopal effect was actually described for radiation, and it's when you radiate one tumor and a distant tumor that didn't see radiation goes away. And it's thought that the reason that happens is that you've induced an immune response at the site you've radiated, and then that immune response is able to work elsewhere. So I think the hint that radiation is actually can have a kind of pro effect on the immune system has actually been out there for a long time, and we're now just learning how to harness that. And a lot of the interesting work going on in this area has shown that if you actually give a slightly lower dose of radiation, it may be better because you're getting that antigen spreading, but you're not killing off the T cells. And I think that's a really interesting area of research now in radiation oncology. We think chemo is similar. So again, it's counterintuitive because we would never do that because we said, oh, the chemo is going to kill the immune cells. But it turns out, one, that's not exactly what happens. It does seem to synergize sometimes. And you may, again, get away with giving lower doses of chemo, which can be more pro-immunogenic. And then, of course, side effects are less.
I was just wondering if there is a second generation of this kind of therapies for tumors that cannot be accessed by directly by surgery. So, yeah. So you get where you don't dose IT, but otherwise.
Yeah. You know, it's interesting because there were two real reasons for going to melanoma early on. One was that you could get to them because they're either in or under the skin, but also they were very immunogenic. But I think now with interventional radiology and endoscopy, there are very few tumors that we cannot get to. And so what we like to say is if you can biopsy it, we can inject it. You know, it just takes getting a needle in there. So using ultrasound or using CT guidance, we can get to almost any tumor. And in fact, part of our development plan at the company is to start with superficial. Once we get some safety there, we're going to inject visceral lesions.
Quick segue to that. Cancer is, I don't know, 300 plus different diseases. You hear different numbers. Any guess from the panel about how many of these will be amenable to this kind of treatment? How quickly this will cascade through? And what about liquid tumors?
I think the hope, so part of what Stephanie showed of the many cancers that are being treated with the checkpoint blockade drugs, that is the hope of immunotherapy, that it is sort of tumor agnostic. As long as there's an antigen for the immune system to target, there is a potential for a response. And so the hope is that this will be broadly applicable.
We've actually studied this in many different cancer types in the animals. So to date, I would say we've done melanoma, lung cancer, head and neck cancer, lymphoma, breast cancer, sarcoma, and we do see activity across all of those. So I don't think there is a priori a reason why we couldn't do this in many different cancers. But the immune system is interesting. We don't fully understand it, and these cancers have unique features that are not always clear. And so it's tricky in terms of development. So I was involved in developing a drug called avelumab, which is one of these checkpoints, and it came up by a company in the middle of a million others. And they said, well, how do we develop this? And I said, well, don't go where everybody else is. Let's come up with a tumor that hasn't been studied. And I proposed Merkel cell carcinoma. It's a really rare skin cancer, but it has a super high, actually a higher mutation burden than any other cancer out there. And so it had all the features of a cancer that we thought would respond well. No one wanted to touch it because there's about 2,000 cases in the United States. It's really, really rare. So from a commercial perspective, drug companies don't want it. And I said, do it there because there's zero competition. And by the way, there's no drugs approved, and it's uniformly fatal disease once it spreads. And if it works there, then you can go in other cancers. And that's exactly what happened. So they got their approval there, and it's now approved in lung and bladder, and they have a gastric cancer approval. And so you just have to start somewhere. And if you get it to, that's why it's really important to get it right the first time, but then you can go elsewhere. And I think the checkpoints are an amazing story where melanoma was first, but now it's approved in 16 different cancers, and including the hematologic tumors as well.
How to further optimize anchored cytokine therapy? For example, can we think of the high throughput screening to optimize 5 factors like one, where, age, cytokine, the dosage, and the duration, 5 factors together in a high throughput manner using spatial omics platforms and molecular barcoding systems?
As far as can we do other things with this, the answer is yes. And, we're focused on oncology for all sorts of reasons, but I don't see why you couldn't extend this to other diseases like infectious diseases. And we are using, we've done a lot of work already with, gene expression, single cell RNA-seq studies. And we're planning to do some biomarker work using multi-omic kind of platforms, and we'll learn about that. And I think this is a perfect platform for exploring the biomarkers so that we understand the bigger picture of what's going on in the patients that we're treating.
I have a question that I think we're going to close out on, but I do want to remind everybody we have a social afterwards. Please interact with the speakers. Please come and ask your personal questions. But my question, I'm kind of targeting this at Stephanie and Dane and Daryl. It's incredibly important for us to connect with our entrepreneurial and also our industry partners, because the only way that our innovation ever makes the way to the patient and to the clinic is through this transition. MIT is never going to be a drug company, and that's why we have these amazing connections. What have you learned about connecting with our industry partners? How is that translation from the innovation to the patient actually fed back to us here to maybe kind of help us focus on how our development pipeline should be more compatible with industry?
You're looking at me.
I am looking at you. Mainly because I know we've been working on a collaboration lately. So
I think it's an instrumental synergy that we have, because MIT, the KI is particularly suited to make breakthrough discoveries and to push the boundaries of what we know and potentially what we can do. And then by collaborating with industry and with clinical partners, I think all have to be in the mix. We can drive the development of drugs, which we're not suited to do, but the industry and the clinical setting is. And I think that back and forth and that open communication actually helps to then find the next boundary that we can push. Daryl, you want to add something?
Well, having gone through a few cycles of trying to take a new technology and move it out, there are two things I've learned are, one, keep it simple, stupid. It's got to be really robust and as simple as possible to see something move out of the lab and successfully get moved into manufacturing and into clinical trials. So very complex technologies, chemistries, they're never going to make it out of the lab bench to the clinic. And then just it's on us as the basic scientists to test these things as thoroughly as we can in the preclinical models, because the company needs to be focused on just moving it into patients. So I feel like our job is to test it as well as we can, understand it as best we can, and then hand it off. And don't leave a bunch of questions unanswered for some company to figure out.
And I'll double down on what Daryl said about the feedback coming back to us as engineers, that it really hones, and carves away foolishness that we might otherwise sit in our clean offices and think are a good idea. Having seen how things move once they go out into the world, I think disciplines us to try and actually do things that can do that.
Can we do better? You get the closing comment.
It's a true partnership. And when I get stuck in the clinic, what I've always done is we go back to the lab. And, when I started my career, we would do stuff in the mouse and take it to the human, but now we're seeing things in the patients and we have to come back to the lab, and we need to come back and ask the questions to the scientists who can get us the answers more quickly. We're learning a lot from patients, but it takes a very long time. And it's got to be a two-way street, and this is how we accelerate, and we're going to get it right. And we're making an impact on cancer. The mortality from cancer is declining across the board for a lot of reasons. It's multifactorial, but there's no question that we have better treatments today, and for the most part, better tolerated treatments. But these are drugs that lead to durable long-term responses. We're not buying patients a few days or a few weeks. We're buying them years, if not potentially curing their cancers.
Well, thank you. Thank you, everybody. Thank you for joining us this evening. We do have another With Insight coming up in early May, where we're going to be focusing on using artificial intelligence and machine learning in early identification of lung cancer. So please join us for that. But in the meantime, please thank our speakers for being here this evening. And we hope to see you again. Thank you.